So last class period, we gave the FACE students this, and now this current FACE students just got this handout. And we said, and so you guys hopefully brought this up and you were working on it at home. You had your digital copy. And if it helps to have the paper one, that's fine. You can decide which one will ultimately turn in. Look at question number 17 and 18. Then come over here and look at here. I asked you guys who are FACE people to put a star by that. Why don't you, current FACE people, do it right now. You got two handouts. Put a star next to codon, anti-codon. And then come over here and answer 17 and 18. You guys at home should practice that as well. Now, let me bring up a couple other things. You know, this generic stuff we went over last class period. If you have DNA and you have to write RNA, the name of that process is transcription. We're going to go through that a lot. Okay. If you're like, well, one part of the quiz is if I just want to practice using the codon chart, number 27, well, you have that, and then you come over here to this, first base, second base, third base. So in a couple minutes, Mr. Matheny is going to just go over different codon charts, just make sure we're on the same page with that. Show you the picture. I'm going to do these models, and then if you need to do more practice with this, number 28 is a good thing to get ready for the quiz. For that part of the quiz. I mean, it's 4 points or 6 of a 20-point quiz. There's going to be a part which says DNA, RNA, or both. Okay? Obviously, we're going to ask you to list the bases. Be a good test taker. Now, on your practice, what you know, you see the bullseye? Last class part, we really only showed you this. So let's, now, to do with this, I need to change a couple settings, so we're going to go ahead and get that situated. And let's see how it works. It's got the blank.
Right, there's a couple charts. You have the chart.
All right.
You have this chart, which we used last class. Figure out your amino acids.
Okay, guys. So you at home, just change the setting, and you'll be.
Whether it be on the SOL or on class stuff, and then you have the side. Right? They're all going to get you the same result, the same exact amino acid. It's just which one you use. Okay. We don't know which one's going to be on the SOL, so we like to show them to you so you know what you're doing. So you're not, you know, what in the world is this? Mr. Moran never showed us this. You know, how to use this. Most of the time, it's going, I'd say 90% of the time, it's going to be a chart like this. Okay? But if you happen to see the bullseye, I want you to know how to use it. Okay.
All right, I just changed a couple settings here. All right, what if you had the following DNA, TTT, that should be a big red flag that you have to do something with that DNA, TTT, before you can use this chart. What do you got to do? Yes. You have to make it AAA. So we're going to use the same 3 letters for 4 codon charts, and if you do it right, you should get the same result.
What is making it AAA? What is that called?
Name of that
What's that
process.
process called? Because really, you could use a one-word answer for that, and it says the exact same thing. Starts with a T. Yes.
Right.
What do we have to do to this? We have to transcribe it. Or what is this process? Transcription.
Right.
Okay, you're absolutely right, Michael. I mean, I was just trying to, again, remember this vocabulary. You're going to see this vocabulary over and over and over.
And this is the, so the details of transcription, molecularly, what actually happens, we're not really going to focus on that. It's some heavy-duty stuff, but I will show you some animations on it. But so 2 words in the chat. One said transcribe, one said transcription. It's the same thing. Transcription is the name of the process. Transcribe is the verb. So when you told him write AAA, he was transcribing. You demonstrating the result of the process of transcription. DNA DNA is transcribed, DNA is replicated. Okay, so here's your codon chart.
Okay, so this is the one we were talking about that you've used already. So we use well, first of all, as Mr. Murray said, TTT. Can I put that into my chart? Why? Why can I put TTT in my chart and get an amino acid?
There's no T in the chart.
Yeah, there's no T in the chart, right? There's no T up here, so that would be like your first catch. Like when you go to put, if you say you forgot to transcribe, all of a sudden you're like, okay, TT, that should be the big warning sign. I missed something, because there's no way you can put a T in there. All right, so we have to transcribe it. So it's AAA. Okay, for this chart, we start on the first base, A. I know it's in this row because the first letter tells me so. First base, it's in this row. The second letter, A. I know it's in this box. Okay, so then I come over here to the third letter and I come all the way across to that box and it's lysine. Okay, it's that simple. So every 3 letters of mRNA is a codon. We plug the codon into the chart. Okay, so this is your first chart. This one's got it all spelled out. Everything's spelled out for you in terms of the amino acids. Second one, the wheel. Okay, a lot of students look at the wheel and go, "Oh Lord, that looks awful," right? What are all those letters? Give me a break, right? But as soon as I show you, I think most kids' reaction is, "That's much easier." Okay, and here's how we use it.
You won't have the will and taste quiz, by the way.
AAA. Okay, the first letter we start in the middle. Second letter on the outside, the next row. Third letter here. And this is how it kind of simplifies itself. First letter is A. I'm only dealing with this quadrant now. It just eliminated all the rest. Okay? So
It's like
I'm going to go
pie.
this way. A. So I'm going to go A, lysine. Okay, same exact thing as our other one. What if I said C, U, G? I go C, it's only this here, U, G, leucine. Okay, what if it was UAC? U, only here now, A, C, tyrosine. Okay, the wheel's pretty easy. Okay, a lot of students seem to like that. Okay, but again, we don't know which one's going to be on your SOL or anywhere else.
On the quiz, we're going to have this available and this available. Actually, I think we have this available as well. I didn't, but you notice the difference? Every amino acid has a full name. Every amino acid has a 3-letter abbreviation. We'll go and show the next one. And we also have a 1-letter abbreviation. We might do something with that the last class before we do our quiz. See if you can spell your name in DNA.
Yeah, whether it's this one or the last one, it's this one. It's still the same principle.
Right.
First base, second base, third base.
Don't be thrown by the letters,
Okay.
the way there's, because there's different codon charts. Remember, it's just a human construct to help you understand. So we'll make sure this works again for AAA.
And so yeah, like this one. Every C, if it starts in C, second letter is a U. It doesn't matter what the third letter is, because all of them are going to be leucine. Okay, which is if Mr. M forwards it to the next one. See how they put the bracket?
Yeah, some students like those because it
kind of narrows it down. These two are both leucine. This one's phenylalanine. Okay, this one's proline. All 4 of these are proline. Okay, so don't get thrown by the chart. They're read the exact same way except for the wheel, which you read inside out.
Yeah.
Okay?
Yeah, so first, second base, left, right, left, top, right. Left, top, right. Left, top, right. Inside, second, outside. If you want to, so we're just giving you a variety of ways of thinking through it. All right? So now we've thrown out a couple terms, and Shane's going to draw something here, and in this, he's going to mention this. He's going to mention this. He's going to mention that. This, that. Oh, man. He has a really good drawing of a ribosome. All right. We'll also talk about the relationship between these, and we might even mention that. Then I'm going to come back, and I'm going to use it with this, okay?
I'm going to mention some other things, too.
So let me get these out of the way.
So we've been talking to you about DNA and mRNA and all this stuff, right? We're throwing a lot of vocabulary at you. We're throwing a lot of processes, transcription, translation. We had you do some practice. We've kind of thrown a lot at you, right? And I don't know about most of you guys, but I'm a very visual learner. Like if I see it, I get it, right? I mean, it's just, let me put my eyes on it and I can figure it out. Okay? So I'm going to make a little drawing here. We're going to kind of wrap up in this picture everything that we've been kind of putting together and talking about. Okay? I'm going to put it into a picture. You can kind of mentally take a snapshot of that picture. When you go to your quiz, you're going to help yourself out a whole lot. Okay? Because that picture will literally tell you what's going on. Okay? And kind of put it all together in a visual. All right? So, and I am no artist, so bear with my inferior art skills. Okay? So, this is a cell. Okay? The whole thing's a cell. Bless you. This for you at home, you can type in the chat too. Mr. Moran's looking at that. What is this here? This circle here? What?
Nucleus.
Good. Nucleus. Why do you say it's a nucleus?
Give me a small circle. And it's the only one there for now.
Not a bad guess, but there's something else that gives it away.
DNA.
Yeah. That right here, this is DNA. DNA is only in the nucleus. It's nowhere else in the cell. Okay? So right there, we've already kind of shrunk it down to where we're starting. We're in the nucleus. This DNA, double-stranded or single-stranded?
Double.
Double, okay? Which makes it big. It's a big molecule. It can't get out of the nucleus. Can't fit out. Which brings in something that we make with that code that can get out.
Yes, but what type? What type, Captain?
Yep, this is RNA. What type of RNA? What letter goes in front of it? No, not tRNA.
Yes, mRNA.
mRNA. Okay, we've got a couple different types of RNA, and all of them have this little letter in front of them that tells you everything about it. mRNA. What's that M stand for? Messenger. What does a messenger do? You're right.
They go out and they download the messenger.
Yeah, they're taking the information, whatever it may be, and taking it somewhere else to drop it off, right? That's exactly what this thing does. When this DNA unzips and we copy, we transcribe it over. Is this single-stranded or double-stranded? Single-stranded, so it can fit out of the nucleus. Okay? We just kind of put it in a smaller vehicle so it can get out. And it's identical match to the DNA. When DNA, we end up producing mRNA. What's this process called? name for it, which we already talked about just a little bit ago. Transcribing or transcription. Transcription only happens in the nucleus. Okay? Does transcription involve DNA? Yes or no? Yes. Good, Michael. Yes, absolutely. Does transcription involve RNA? Yes, absolutely. That's what we're making. But we're making it from the code of the DNA. All right? So we kind of got a little bit going on here. That's our first step. All right? And it's all in the nucleus. Transcription, DNA, mRNA, messenger. So that messenger is going to slip out the nucleus. Okay? And it's going to get out here in the cytoplasm. And it's going to look for a what? Starts with an R. That makes proteins. Ribosome. Beautiful. It's going to go out and look for a ribosome. Okay? It's going to go look for a ribosome. Okay? Because it's carrying the code, right? It's like the one of those Brinks armored trucks driving from one place to the other. They got all the money in it, right? They're waiting on it. Bring it here. Bring it here. Here's our ribosome. Here's our mRNA. What is every 3 letters on mRNA called? Those 3 letters. What are these 3 letters called if we put them in that 3? Starts with a C. Codon. Right? Remember the word codon? Okay? Codon. This is a codon. This is a codon. This is a codon. Codon, codon, it just keeps going. All right? And that's important. Okay? Every 3 is a codon because we have this other thing out here that's coming down to match it. Okay? Kind of looks like a little spaceship. It's got this thing hanging off of it. This is a tRNA. Okay, tRNA. Do I know what the T stands for? Yes. Beautiful. Transfer. What's it transferring? What is this thing right here that's hanging off of it? Starts with an A, and the second word starts with an A. Amino acid. Yep, amino acid. Okay. How does that tRNA, that little spaceship, know to drop this off? Where to drop it off? It's got a little code on the bottom. Okay. This is called, anybody know? Anti-codon. What's anti mean? The word anti. If I'm anti something, I'm not for it, I'm against it, or opposite. Okay. This anticodon has to match the codon perfectly, or it will not drop it off. Okay. So, for example, what if this codon was CAU? That codon was CAU. What would match the C in RNA? What goes with C? What goes with A? U. Good. What goes with U? That would be the anticodon. CAU is the codon. GUA would be the anticodon. If it doesn't match perfectly like a key in a lock, doesn't drop it off. Okay? But for our sake, great, it matches. Okay? So it's going to drop that amino acid off inside this ribosome. Then the next tRNA comes down, and it's going to try to match. And it's going to drop off its amino acid, and its amino acid, and its amino acid. It's going to keep going. Okay? When we have a string of amino acids together, that makes a what? When you put amino acids together, you get a, yeah, protein. Absolutely. You get a protein. Isn't that the whole point of what we've been talking about? Protein synthesis. Protein synthesis, to synthesize means to make. So we are making a protein. Where does it start? All the way back here. Okay? So we said this process of making mRNA is transcription. What is this process called? I can't tell if they're typing something good or not. Translation. Good. This whole process is called translation. This is where you're plugging your letters into your chart. Okay? This is where you're finding out what amino acid goes. This process, you're plugging in. This is where we pair up the bases to make RNA on your paper that we did last class. Okay? Then when I plug it in the chart, it's here. Okay? So kind of gives you a little bit of a visual. Again, it's kind of ran through a lot of our vocabulary, okay, that we've used so far. Any questions? Again, try to capture this picture in your head. So when you get on that onto a question, no matter what it is, where does translation take place? It's not at the tRNA is involved in but where? Like what place? At the ribosome out here in the cytoplasm, right? There's a bunch of ribosomes all throughout in the cytoplasm. So yeah, cytoplasm. Oh, where's transcription take place? Man, Mr. Matheny wrote transcription down here, so it had to have happened in the nucleus, right? So use my picture to help you. Use my picture to kind of spur your thinking and eliminating answers as well as finding the right answer. Okay. Any questions? Anything? Hopefully this helped because again I'm a visual learner and that makes puts it all together for me. Hopefully at home that worked as well. Hopefully you guys got something out of that and it will help you on your quiz.
All right, so let's do some models, and so just take a second. Anyone at home, if you want to do it, you're welcome to do a little screenshot of that. We are recording this, and I will send it out. And one of the things I'm trying to also do is all right, we're going to go to the models in a second, but I want to bring in, I have the right one. No, I don't have it. Right. So, all right, I'm going to share that second. Let's do this. Let's go to the other models here. So we still have camera. Mr. Matheny, can you erase that real quick?
Yep.
Okay? So I'm just going to write here. Hopefully this works. Okay. Am I in there?
Yep.
Okay. Hey, what is it?
What T is that? What is it?
And also, you guys who are at home,
DNA, right? How do you know that's DNA?
you're going to need a codon chart.
Yeah. There's a T, right? There's a T. It's got to be DNA.
Okay, so we know that that is DNA, right? Okay, good. Where is it? Where is this DNA?
Think about it by a picture.
It's going to be in the nucleus, okay? As opposed to cytoplasm. I'm going to put the cytoplasm over here. Then later I'm going to move this out and put the DNA over here. So, hey, what goes to T? What goes to T? What goes to A? T. Now, at this point, if you just said T, you could have said A and be DNA or RNA. Now we need to clarify. Let me slow down. Now we need to clarify and say, what's the complementary strand? Or what's the DNA pair? Then you would have been correct by saying T here. But if I said, what's the RNA, what would you have put here? Yeah, so let's do the DNA first. Okay, so DNA, all right, look at this. These are the rungs of the ladder. Rungs, these are the steps of the ladder. But what is not pictured here should be the sides of the ladder.
What makes up the sides of the ladder? Huh?
This is the part where
Well, it doesn't matter if it's RNA.
This.
It does matter. The
Is
sides
that?
of the ladder.
I'm talking these.
There's 3 parts to a nucleotide. 2 of them make up the sides. 1 of them makes up the rung.
Yes, deoxyribose, which is a sugar, and
Say it louder. Yes.
phosphate, yes.
Sugar phosphate, over and over and over, all the way down the side and all the way down the other side. And even on RNA, which is single-stranded, that whole side is sugar phosphate. Now, the sugar is different. The sugar in DNA is deoxyribose. That's where the D comes from. The sugar in RNA is called ribose. That's where the R for RNA comes from.
Okay, now, what bond between these bases am I just breaking? That didn't sound right. What bond did I just break? The hydrogen bonds. What bond am I now forming? Hydrogen bonds again. I should have a card for hydrogen bonds, but okay. The name of this process is, you just did it. When we said T and you wrote AAA, that had a name. What is it? Transcribe or transcription, all right?
You're right.
So this is transcription in action. When you write each letter, you're transcribing. So it's the same term. Don't be thrown by the language. Okay. So once the messenger RNA is made, this is going to zip back up because DNA is constantly zipping and unzipping. So I'm going to move my nucleus over here. Oh, look at this, like a nuclear membrane. Okay. So now, I don't know if you guys can see it over here. What we should say is, this was made here, and now we're going to go. I'm going to take advantage of my cord. So imagine this being the nuclear membrane. It's coming out, it's coming out, and it's going to come out, it's going to come out, and it's going to stop at a ribosome. So I'm going to put this down here. By the way, let's get some other things here that we're going to be talking about. Leave that over there. Oh, that's what the rRNA is. This is the mRNA. So I'm going to move my cytoplasm up here. Oh, DNA should be over here.
The R in rRNA, the small r, stands for what? Mr. Mary said? Is that ribosomal RNA, right? So that little letter tells you exactly where it is, what it's doing, all that good stuff. Messenger RNA, transfer RNA, ribosomal RNA.
Boom.
Tells you exactly where it's at.
Transfer RNAs. But the transfer RNA has an amino acid on it, but I'm not going to put it there until you tell me. Okay, so let's do this. Does that pair? No, it doesn't. We still have to do base pairing. What goes? All right. So before we translate it, what goes to A? What goes to C? U? What goes to this? I think I did grab the right one. Okay. Now, the problem is there's an amino acid that's going to be there. So what? How do we know what that amino acid is? We now have to translate the thing we transcribed. Think about it like that. That's the order. Use your codon chart. So you need to tell me what amino acid is going to come in by the messenger RNA. So we would DNA, the RNA in the nucleus. RNA goes to the cytoplasm, hooks up on a ribosome. Now tell me.
AUG.
What is it?
What's that in your chart? Thymine.
Thymine.
Good.
So watch what happens. It kind of clicks in. When I say click, it's base to base. What type of bond is that? Hydrogen bond, but this time the tRNA is carrying in. So I got tRNA, but let's do this.
You guys haven't figured out, if he asks you a question about a bond, it's the only bond we've talked about for 2 weeks.
Yes, we're going to save one more today, but hey guys, if you're still trying to figure out codon, anti-codon, oh. And if you want to be technical, remember a tRNA, so here's the tRNA, it has two parts, the anti-codon part and the amino acid part. That make sense? Okay, so I'm going to leave this here. I'm going to move this out of the way. Just leave this as my all floating tRNAs. Okay, we got to translate. We got to translate. Translate the transcript. That's another word of calling RNA. So GGC. So we know the hydrogen bond is going to be with what? CGG. Now again, sometimes students make this mistake. They go DNA or RNA, and then they go to the tRNA, and then they stick that in the chart. Do not do that. Once you have mRNA, use it in the chart. By the way, that chart's called a codon chart. That chart is an mRNA chart. Okay, for our purposes. So someone tell me, translate it. Use the chat. Also, tell me what is this? What is this tRNA going to carry in? And you know by looking, translating the mRNA with the chart. First base, second base, third base. Come on, people taking a quiz.
What is GGC?
Glycine. Now watch this. This comes in. Now I'm going to do this. This tRNA is going to come over to tRNA land, which is basically floating around in the cytoplasm. It's going to pick up another methionine and wait till it recognizes a signal on a ribosome that there's an RNA. I need to drop this off. It doesn't say it. It doesn't have lips. They don't actually talk. All right. Hey, all right. We have our second amino acid, AGU. All right, AGU, which is going to be what, UCA for the tRNA. But you need to tell me what amino acid is going to come in. Translate. Serine. Okay, hey, looks like we got another thing to translate. GUC. So tell me. Valine. By the way, valine is one of those ones that has like 4 combinations, but it's usually the third base that's the one that could be variable. Okay. All right. All right. Hey, I got one more codon in the messenger RNA that was transcribed from the DNA. So now translate the last one. Use your chart. Stop. Remember, there are 3 stop codons. UAA, UGA is another one. So what that does is it says that. That's what the stop does. This goes to pick up another valine for the next time it needs to do this process. Now, let's one last bit of label. I'm going to bring in a bond you haven't seen in a while. The yellow thing is a peptide bond. Won't be asking it today, but that's, remember back when we did chemistry? This is an amino acid, and the whole thing is a protein. All right, I know what's going to happen. As long as there is tRNAs back, the next round of another ribosome come through and translate and produce another one, and then another ribosome is going to come in. As long as these are in the cytoplasm, picking up the amino acids, you can mass produce it. This is how your body mass produces the protein for a gene you need. In your body right now, if you needed to produce insulin because you had some carbohydrates, this process was going on. And imagine these being the amino acids that fold to become protein, the protein that we call insulin. You can synthesize about millions of them in a couple seconds. That's how fast this process is happening. And it all starts with the signal of the DNA to make that RNA. All right. If you want to know the expanded term, this is what is referred to as gene expression. That's not invasive. Okay, I'm going to do one thing here. So I'm going to, I have to switch it so you guys at home can see, or no, the space students can see. And I'm going to, here. How are we on time?
It's 9:08.
They're out.
What, 9:40?
Okay. All right, so guys, this is just here's DNA. There's DNA unzipping. Here's DNA making RNA. And then you go to the cytoplasm where this happened. So, all right, that's the models. I think we're pretty good. So let's do this. I'm going to stop sharing that. As we get ready for our quiz, you FACE students need to go here. There's this right here is a PDF. Actually, everyone at home can open this. Open the PDF. This is the PDF. That's the codon charts that you're going to use on your quiz. All right.